Hello viewers. Today, we have some exciting news to share about a groundbreaking development in treating type 1 diabetes. We will dive into the details of a new islet cell transplantation therapy showing incredible promise. So, let's get started. The breakthrough. Hypoimmune islet cell transplantation, Sana Biotechnology, has recently announced positive clinical results from their study on islet cell transplantation for type 1 diabetes 1. What makes this study so remarkable is that it achieves something previously thought impossible: successful transplantation without the need for immunosuppression. What are islet cells? Before we go further, let's quickly recap what islet cells are. These are clusters of cells in the pancreas that produce insulin, the hormone crucial for regulating blood sugar levels. In type 1 diabetes, the body's immune system mistakenly attacks and destroys these cells, leading to insulin deficiency. The challenge with traditional transplants: islet cell transplants face two major hurdles: the need for immunosuppression drugs to prevent rejection, a shortage of donor pancreases. These challenges have limited the widespread adoption of islet transplantation as a treatment option for type 1 diabetes. How does the new therapy work? Sana Biotechnology's breakthrough lies in their hypoimmune HIP technology. This innovative approach involves genetically engineering the islet cells to make them invisible to the patient's immune system. Here's how it works. The cells are modified to avoid both allogeneic from a different person and autoimmune detection. This engineering concept, called HIP CD47+, can be applied to various types of islet cells, including those derived from stem cells or isolated from human donors. The result? Transplanted cells that can survive and function without triggering an immune response, eliminating the need for immunosuppression. The clinical trial results now. Let's look at the exciting results from the clinical trial. Insulin production. The transplanted cells began producing insulin, with the patient showing decreased insulin requirements as early as 2 weeks after the transplant. Insulin independence. By day 75 post-transplant, the patient achieved complete insulin independence, which was maintained for at least 12 months. Blood glucose control. Fasting blood glucose levels improved dramatically, dropping from an average of 210 milligrams per deciliter before surgery to 126 milligrams per deciliter after. Time and range. The patient's time in the target blood glucose range increased from 43.18% to over 98% during the 8 months of follow-up. HbA1c levels. The patient's HbA1c, a measure of long-term blood sugar control, decreased from 7.57% at baseline to 5.37% by day 120, falling below the diagnostic threshold for diabetes. These results are truly remarkable and offer real hope for a scalable, potentially curative treatment for type 1 diabetes. Advantages over traditional islet transplants. Let's compare this new approach to traditional islet transplants. No immunosuppression. The biggest advantage is the elimination of immunosuppressive drugs. This is a game changer, as these drugs can cause serious side effects like increased risk of infections and kidney damage. Potentially unlimited cell source. By using engineered cells, this approach could overcome the shortage of donor pancreases. Improved graft survival. Early data suggests that the hypoimmune cells may have better survival rates than traditional transplants. Long-term efficacy. While more data is needed, the initial results show promise for long-term insulin independence. Potential risks and challenges. While the results are exciting, it's important to note that this is still an experimental treatment. Some potential risks and challenges include procedural complications. As with any transplant, there are risks associated with the surgical procedure itself. Long-term effects. We still need more data on the long-term effects of these genetically modified cells in the body. Regulatory hurdles. In the United States, islet cell transplants face regulatory barriers that could limit access outside of clinical trials. Scalability. While this approach could potentially solve the donor shortage issue, scaling up production of these engineered cells may present new challenges. The bigger picture, a new era in diabetes treatment. This breakthrough is part of a larger trend in diabetes research focusing on cell replacement therapies. Other notable developments include stem cell-derived islets. Researchers are making progress in creating insulin-producing cells from stem cells, offering another potential source of transplantable cells. Autologous transplants. A study at Peking University successfully treated a patient using islet cells derived from the patient's own cells, potentially eliminating rejection issues. Alternative immunosuppression. Other researchers are exploring new immunosuppression strategies that may be less toxic to islet cells. These advancements collectively represent a significant step forward in the quest for a functional cure for type 1 diabetes. What this means for patients for people living with type 1 diabetes. These developments offer hope for a future where daily insulin injections may no longer be necessary, blood sugar levels could be naturally regulated, the risk of diabetes-related complications could be significantly reduced, quality of life could be dramatically improved. However, it's important to temper excitement with realism. While these results are promising, more research is needed before this treatment becomes widely available. The road ahead. So, what's next for this groundbreaking therapy? Larger clinical trials. The next step will be to replicate these results in larger groups of patients. Long-term follow-up. Researchers will need to monitor patients for several years to ensure the treatment's safety and efficacy over time. Regulatory approval. The therapy will need to navigate the complex regulatory landscape to become an approved treatment option. Accessibility. If approved, efforts will be needed to make this treatment accessible to all who could benefit from it. Conclusion. The development of hypoimmune islet cell transplantation marks a significant milestone in the treatment of type 1 diabetes. It offers the tantalizing possibility of a future where insulin independence can be achieved without the need for lifelong immunosuppression. While we're still in the early stages and there's much work to be done, this breakthrough provides real hope for the millions of people living with type 1 diabetes worldwide. It's a testament to the power of innovative research and the tireless efforts of scientists, clinicians in the field of diabetes. As we move forward, it's crucial to continue supporting diabetes research and staying informed about these exciting developments. Who knows? The next breakthrough could be just around the corner. If you want to stay updated with new diabetes developments and research, you know what to do. Thank you for tuning in to our diabetes channel. Remember to stay hopeful, stay informed, and most importantly, stay healthy. Until next time, if you like this video, please don't forget to leave your comment below and show your support by liking, sharing, and subscribing to our YouTube channel. Your support means so much to me, and it helps me continue bringing you valuable content. Thanks for watching,